Started with our presentations tonight. It is student night, student research night, and I'm really excited. We have four researchers, one from Cal State Fullerton, two from Cal State Long Beach, and then one from UCI. All really great. Some local stuff and then some other stuff that is also really great to hear about. So our first speaker tonight is going to be Tayahna Agtarap. Her bio, Tayahna is a recent graduate from Cal State Long Beach. She received her BS in biology with a concentration in molecular and cellular biology. She is conducting research in Dr. Amanda Fisher's lab, investigating the relationship between pollen-ovule ratios in pollinators across the genus Justicia. She plans to pursue her PhD in plant molecular biology to better understand plants' responses when exposed to abiotic stressors. So with further ado, I am going to ask Tayahna to come up here and present her research. Also, I will say it's going to be about 10-15 minutes per presentation, and then we will follow with about a little bit of a 5-minute question time. So if you have questions, wait till the end, and we'll call on you. And yeah, thank you.
Hello, everyone. Thank you for coming and thank you for that introduction, Mel. My name is Tayahna Agtarap, and my project in Dr. Amanda Fisher's lab focused on investigating the relationship between pollen production and pollination methods in Justicia. So, previous research has found that flower gamete production is related to reproduction efficiency. And plants and pollinators, we know, have a mutualistic relationship where plants provide food in the form of pollen or nectar to the pollinators, and in this exchange, pollinators move pollen from flower to flower, fertilizing the ovule. We also know that plants have evolved various mechanisms in order to optimize their reproductive success. We've seen this through plants that have various colors, shapes, and sizes of their corollas in order to attract various pollinators, and this can be seen here through these different species of Justicia here. So one thing we know is that these flowers are able to attract different pollinators through their different corolla shapes, size, and colors. But we wanted to know if pollen quantity was something that helped a plant optimize its reproductive success as well. So one thing that my project is based on is the idea of this relationship between plant pollination efficiency and the pollen to ovule ratio, or how much pollen that a plant produces. And so the idea is that if a plant has high levels of pollination efficiency, it would have a low pollen to ovule ratio. And we would expect that the opposite would also hold true, where if we have a plant that has low pollination efficiency, such as something that is wind pollinated, because there's pollen being lost in that transfer from flower to flower, we would expect that there would be a high pollen to ovule ratio to compensate for that loss of pollen. Now, to give you some background on some of the species that I was sampling for this project, you've probably seen this going on hikes around Palm Desert. This is Justicia californica, and it is the only Justicia species that is native to California. It's also native to southern parts of Arizona and northern parts of Mexico. It is found as a flowering shrub, and it is also hummingbird pollinated. So again, this is Justicia californica. Below here are the anthers of it, and then an SEM image of the pollen. The rest of the species here that I studied are Justicia botanica and Ayers, which are bee pollinated, Justicia californica, Kansas sonori, which are hummingbird pollinated, and Justicia lithroides and longii, which are lepidopteran pollinated. And it's pretty amazing that all of these closely related species within the same genus have such a wide variety of pollinators. And so I'm studying these species because I wanted to see how pollen to ovule ratios evolve among closely related plants. In this next row here, my project is also based on the different morphologies of the anthers. Here you can see in these left two species that there's kind of this anther extension, and that is something that my project is based off of, and that's also based on previous research done by Dr. Keel, where she found that these different morphologies in the anthers, where you can kind of see how some anthers are positioned right next to each other, some are kind of offset from each other, and she found that they are correlated to different pollinator groups. Next here, you'll see SEM images of the rest of the species. And the Acanthacea family is very famous for having different pollen textures, shapes, and sizes. And my lab mate Valerie found that these differences in the pollen are also correlated with different pollinators as well. So that leads us to my hypotheses. My first one being that flowers of Justicia species with different pollinators will produce different amounts of pollen. My second one is flowers of Justicia species with beaky extensions, like I had previously mentioned. They have evolved these beaky extensions for, with a new function to scrape pollen off of pollinators, which we would have hypothesized that they would produce less pollen in those beaky than flowers without those beaky extensions. So for staining and counting the pollen grains, yes, I counted all of them grain by grain. It started off with me dissecting each flower. I dissected each flower, took out the anthers, and then I separated them along the filament in order to separate the four beaky. These are the anthers, and then individually I refer to them as beaky, singular, beaka. And on the right here are the ovules. For my project, we wanted to make sure that all of the samples had 4 ovules, which they did. And then I stained the pollen grains with an acetyl-orcine stain. And orcine is a pigment that is derived from archaeolichen, and it kind of has this pinkish red hue to it, which stains the pollen that same color. I placed the pollen grains in a Raptor cell and counted them underneath a compound microscope with the help of my lab mates Kaya, Daniel, and Kylie. So that leads us into my results. For the first set of results, I found that Justicia species with different pollinators produce different amounts of pollen. And on the x-axis here, I have the different species, and on the y-axis, I have the amount of pollen that was produced, or that pollen to ovule ratio. And then they're also color coordinated according to their pollinator. We did find that they were all significantly different from each other, according to their pollen to ovule ratios, except for the ones that are denoted by the letters here. Which are Justicia botanica with Justicia californica, Justicia pedularis and snorri, and then Justicia lithroides and Justicia longia. We are familiar with some of the reproductive biology of some of the species, and this was done through previous research. The first one being for Justicia botanica. Previous research found that it is an obligate outcrosser, which means that it is required for the pollen to come from another flower in order to be successfully pollinated. And so this is consistent with what we were expecting, because since it is an obligate outcrosser, we would expect that it has high levels of pollen in order to compensate for that loss of pollen. Now, these next species that we do know for the reproductive biology that we're familiar with, we have found in previous literature that it has a mixture of selfing and outcrossing. Now, again, it's also consistent with what we were expecting. Because it outcrosses, it still has those high levels of pollen to ovule ratios, but because it is able to self when needed, it's not quite as high as Justicia botanica, which only outcrosses. And then for these remaining 4, we are not entirely familiar with what their reproductive biology is. But this brings us into grouping the species according to what pollinates it. And we found that Lepidopteran pollinated species produce significantly less pollen than those that are pollinated by bees and hummingbirds. This moves us into the second hypothesis that I had. So if you remember, I mentioned those beaky with that little hook or that extension at the in the beginning. And what my hypothesis was that the ones with that extension would produce less pollen than the ones without it. However, what we found was that it actually produces more. So in this diagram here, I found that when we compared all of the inner thecae compared to all of the outer ones, we didn't see a significant difference. However, when we compared all of the thecae, when we have an extension and when we don't, again, we found the ones that had that extension produce significantly more pollen. So again, we found that Lepidoptera and pollinated Justicia species produce significantly less than bee and hummingbird. And this can suggest a variety of things, like Lepidopterans may just be more efficient pollinators because they produce such little pollen that the plant doesn't need to kind of compensate for that. Or there might be higher rates of selfing because since it is able to self-fertilize, it doesn't, or self-pollinate, it doesn't require such high amounts of pollen. There are a lot of things that contribute to the reproductive success of a plant, and so we're not entirely sure which one it is, or which one can be responsible for it, but that definitely is something that we are looking to do in the future. And the second hypothesis was that the fungi with anther hooks produce more pollen than those lacking them. And again, this was extremely surprising. But one thing worth mentioning is that the species that had these anther hooks are also bee pollinated. And if you remember from the previous graph that I showed, they produced such high amounts of pollen being bee pollinated. So we're wondering if this is because they are bee pollinated, so they're less efficient pollinators, which is why they produce so much pollen, or it could just suggest that there is that reduced efficiency in the bee pollination. And so our future directions, we would like to sample more bee pollinated species to further investigate the variation that we observed when we were counting, when we saw the pollen quantities. Our second one being that we want to sample species with varying anther extension lengths because we have some species that have that full anther length or the full anther extension and then some that are a little shorter. So we wanted to see if they would produce different amounts of pollen. And then our third one would be to assess those selfing rates in Lepidopteran pollinated species, just like the previous slide that I was showing where we weren't sure what was responsible for that low amount of pollen that we saw. And so we were wondering, is it because of that pollinator efficiency that it produced so much or such small amounts of pollen, or is it because it's selfing? And I'd like to thank the Cal State Long Beach Rice Program, Dr. Amanda Fisher for all of the guidance through my research and this project. The rest of Dr. Fisher's lab, Brian Thorson, our Cal State Long Beach Botanics greenhouse technician, Dr. Carrie Keel, and Orange County, California Native Plant Society for having me today. Thank you so much. I'm glad to take any questions if anyone has any.
I have a question.
Yes.
Is there gonna be, is there a difference between like the perennial versus, I don't know if there's any annual justicia, but is that something to look out for like within this at all, like with their characteristics?
That's a really good question. To be honest, I'm not entirely sure, but that's definitely something that we can look into because that was a good point. Thanks. Yeah.
So do you think like locality or like habitat could affect whether or not there's more or less pollen? It's like in a more desert environment, they might not want to put out that much pollen.
Right, yeah, no, that definitely makes sense. Like if it requires more pollen than in a specific habitat, that would make sense. There's just so many, it's like this tangled web of all of these different factors that factor in. So why like certain species produce different amounts of pollen that you would have to like, I don't know, kind of test every single one to like weed everything out. But yeah, that's definitely a factor. Yes.
The lepidopteran pollinated plants produce so much less pollen. Is that true in other plant groups, or is that unique to the Justicia?
I'm not sure if it's only unique to Justicia, but I have found that, I think I found a paper where it was actually saying that the Lepidopteran pollinated species produced more pollen than the other ones, yeah. So I don't know, I'm not entirely sure what other species produce that have, significantly less pollen, but it's the Lepidopteran pollinated, but yeah, that's a great question.
Cool, thank you so much, and then we will go to our next speaker, so. Cool, so our next speaker is Ayers. Anna is a recently graduated undergrad student from Cal State Fullerton with a degree in ecology and evolutionary biology. Her ecological focuses are plants and amphipods. She worked in Cal State Fullerton's McFadden Herbarium with Joshua Durr for 4 years, assisting in imaging and digitizing the main collection. In addition, she worked in Dr. Christy Corbin's lab, working at the reproductive systems of fish. When not focusing on ecology, Anna likes to be an unprofessional chef at home. Pull your mics up there.
Hi, I'm Ayers, and I was my project on adoption trees, specifically, niche modeling for adoption trees.
Yeah, hold the microphone.
A little bit closer.
Higher?
Yeah, there you go.
Awesome. I'll fix that. So specifically for the niche modeling for adoption trees. So when I decided to do this project, I didn't really know exactly what species I wanted to do my research on. So I was kind of sitting there thinking, what's a cool species I like in California? And my professor kind of kept hinting on Joshua trees. So I spent like a couple of months doing a bit of like a literature review on them. And I just went on Joshua trees because it seemed like they had a lot going on with them. I was looking into them and I saw that they, it takes a while for them to fully mature. They don't mature until they're about 20 years in age. And if you're a tree that's facing like an immediate threat, you don't want to wait 20 years to actually fully bloom and bring more seeds for their trees. It's just not a good combination. They're also really good important species for their Mojave environment. They're keystone species. And along with that, they're keystone species, they have an opposite mutualism with a specific moth, the gecko moth. So these species, they can't actually pollinate without this one moth. So again, they're kind of handicapped in that sense. They don't have a lot going on for them because they're kind of fully reliant on this moth, but they also provide a lot of, they provide this plant for the moth too, which is also very important. They have a variable germination rate, so it's kind of hard to fully determine how they germinate. I was reading in on them and it was really discussing how when they flower in like big bunches, but it varies, so it's gonna be like, all the trees are all gonna flower around the same time. But it's not gonna be the exact same time every season, it's not gonna be every year, sometimes it's twice a year, sometimes it's every other year. It's just very variable based off of the environment, and being in the desert, it's not really the best environment for them to flower a lot. And the fact that they have a variable germination rate doesn't help the fact that they have a low seedling establishment. So the seedling establishment, it mainly depends on the seedlings blooming after kind of resurfacing after lots of rain. And if they're in the desert, then it's already hard for them to really bloom, to have the seedlings release. And they have to have a lot of rain for the seedlings to fully like reach through the surface. It's just not gonna be good for them. So Joshua trees, they don't have a lot going on for them. So we have to see what's going on for them. Going on to this, there is currently a Western Joshua Tree Conservation Act. So they're not an endangered species, but it's just a protection act to make sure that they are being protected while they're trying to be like considered endangered in the meantime. So this act is mainly focusing on the Yucca brevifolia species. That's what we have here in California. They're the Western Joshua trees, so they're the bigger guys. I heard from some people who are in different states like in Arizona and Nevada, that's where they have the smaller eastern species. That specific species of Joshua trees, they're not as endangered, so they don't have a conservation act for them out there. But focusing here in California, we have our Western Joshua Tree Act here. This act, it prohibits any unauthorized import, export, take, sale of Joshua trees. And it also requires California Fish and Wildlife permits for any incidental takes or hazard levels. So if you have the Joshua tree, it's going to be removed if it's seriously impeding on its area. And it's also going to be developing a conservation plan, which I'll talk about a little more. So I work at an herbarium, and this project involves herbarium specimens. And this is an herbarium specimen here at the Big McFadden Herbarium. So we haven't seen any of these things before. This is what they look like. Just a bunch of dead, dry plant species or samples. And the main reason why we're using the herbarium specimens is because each specimen has a little label. And on the label, we have a little information on them. It involves their elevation, the date they're collected, the collector code, and usually depending on the specimen or who collected it, it also have coordinates. So for this project, I was mainly using herbarium specimens, so we could take their coordinates, elevation, collector code, like the date and all that. So we just make a niche model map for them. When I was making the niche model map, I took different samples from iDigBio, and it's just this really nice like database that has all these digitized specimen collections online. So anyone can basically access this information. If you want, you can also make a niche model map at home, very easy, all this is free. And so I downloaded all of my information from iDigBio, and I had to clean up the data, so I just had to make sure all the taxon names are resolved. There are some issues with what is an eastern Joshua tree, what's a western Joshua tree, so I had to fix that up. We had to remove duplicates, so sometimes there could be one species listed under like one person's name, then all of a sudden 5 names down, it's a different person's name, but it's the same coordinates, so we just want to count it as one tree only. And at the end, I only had 208 data points. And this is for both the western and the eastern Joshua tree, which honestly is not a lot, but we are working with what we have. And after I got all those data points collected, I went through my climate layer processing. And I got all my climate layers from WorldPlan, and I had to remove all the correlated layers. So basically, this means if I have a lot of layers, if a lot of them seem to be impeding on the trees, or if it doesn't really seem to impact the trees or any of those points, we want to remove them. That way, whenever you produce the map, it's not going to have any like weird implications. I'm not too sure how to best explain this, but the layers that have the most impact on the trees were these layers right here. So it's gonna be the diagonal range, the isothermality, annual temperature range, the wettest quarter, coldest quarter, precipitation, warmest quarter, and precipitation levels quarter. And then elevation did not have any correlation, but we decided to include it anyways, just because the herbarium specimens also included elevation in their points. And then we had to go over the good and the bad emission scenarios, because we're trying to figure out how these trees are gonna be in the future. And so in the future, we're gonna be having an issue with our carbon. So under a good situation, we have our optimistic scenario, which is basically from this day forward, every single person is gonna do their part in kind of reducing their carbon export. So that way there's not gonna be a lot of carbon, the world's gonna get better, things will be good. And then we have our bad scenario, which is actually the situation in which no one does anything, it's business as usual. No one's gonna do anything to kind of help the carbon emission rate, and it's just the worst case scenario. Which is a little worrying because it's also basically what we're doing right now, but whatever. And I got all these carbon emissions from the HAGEM, which is basically it's the British producer for these carbon emission scenarios. And I have a lot of maps, so at the end, I will have to flip back and forth to look at everything. But all these white points right here, the little squares, these are all the data points for the Joshua trees, and these points are gonna be in the same location for each map, just so you know. So on the left, we have the Yucca vermifolia, the western Joshua tree, and on the right, we have the Yucca Agariana, the eastern tree. And this is the historic data, so basically, this was their preferable distribution. So if it's in an area that's warmer, so anything that's red, that's where it would be the happiest. And if it's in the blue area, it's not gonna be so happy there. So a long time ago, they used to have all this area where they could be happy. And then now we're looking at the good scenario in about 20 years. And I want to say it's a little bit different. Under the good scenario, we already have a greater reduced distribution range. And as you can see, these are points where you found the Ariana, they're not gonna be in a good location. They're not gonna be happy here, basically in about 20 years. They're gonna be up here, and again, same over here. None of these guys are gonna be so happy here. And that's the good situation in about 20 years. And in about 40 years, it changed a little bit. Like it's kind of weird because as you can see here in the Ariana, they have a preferable range where they previously were not preferable. And the best kind of way to explain this would just be in about like that many years, it could be warmer in that area or it could be a bit more wet in that area or in that specific elevation location. it just makes more sense, but we don't know exactly why they're gonna be there. But overall, all the elevation or the preferable distribution is being reduced over time. And in the bad scenario, it changes a lot too. So again, if from Yadkin Revifolia on the left, they're not in their preferable location. And Agariana is also not really in their preferable location. And over time, you kind of see that their preferable location is being pushed. So Revifolia location is being pushed a bit more northwest. And then Agariana is also being pushed a bit more towards the north and towards the east. So they're separating a lot more, which is a little interesting because they're different species. And some people also still don't believe they're different species, but there's kind of like arguments like why they're different. They have different places they prefer. But again, in about 40 years, Jeff and Florida, they're not in preferable locations unless you have free Gariana. So what does this mean? Basically, in all these scenarios, there's a decrease in superhabitats for Joshua trees for both species. And with climate change, it's gonna be very impactful on Joshua trees because they don't have a good distribution range. And their preferable distribution range is being drastically decreased. And you can always think, okay, so they're not a good distribution range, now what? Well, they also don't have a good distributor, so it's not like they can just move, they can't walk away their trees, and they kind of germinate to preferable places. And then they also they have so much going on for them. Which is why it's kind of important that we have our conservation plans. And I mentioned before for the Western Joshua Tree Conservation Act, they have a plan, and a part of the plan, they have to have an agreement that says that they are gonna be looking into assisted relocation for the trees. And they're specifically looking into doing this relocation with different native tribes. So that way they can determine which area is best suitable for these trees for ethnobotanical reasons. But it's also just something that people should be aware of. This is just Joshua trees, but this could happen for any other species. And that's it. Any questions?
Okay, I had a great talk, by the way. I had a question for like some of the areas that have like red in like 40, 80 years. Are like some of those areas already preserved and like, is it like the Mojave National Monument or something or?
We don't know, well, we know that this area, we're pretty sure this is the military base for like, for location. This area, we don't really know what's going on. We were trying to figure out like where everything was, like on the map, like this is California. That's Nevada, Arizona, but we're pretty sure this is like a mountain, and the only reason why this has a different preferred location versus everything else is because of the change in elevation, that's what we were saying. But we know for sure that this area is a military base. So it's probably gonna be preserved over there because no one's gonna be going into military base, but also, yeah.
Have you looked into seeing if there's any Arcadia data about like the very historic range of the species?
I haven't, but I read a bunch of papers that were talking about that. And the main reason why they have such like a more preferred distribution for the Banuelas, I think it's because they had a different distributor or, they had a better distribution range, historically, because they had an actual distributor, and they had like a giant ground sloth that carried the seeds around, I love that guy. So that's why they had like, it was easier for them to actually get to these locations, basically, because they had a big guy who could take them to these places. But I'm not too sure how that actually impacts their preferred range, like what about that historical climate made it easier for them to grow.
So there's a big project in Kern County that was approved. And they're destroying thousands of trees for solar panels.
Yeah.
So how does that fit in with the conservation plan?
I was reading into it and honestly, like. It seemed like that. I guess technically, like, what they could do is a part of the conservation plan, it was one of the, what was it? Allowed, removal of the trees, technically, basically, if they have a lot of money, they're allowed to have an incidental take, to take the trees out. As long as that money goes into funding for the act, which seems a little contradictory, but that is how they decided to allow that.
So no, Joshua trees on public land are protected, or Joshua trees on private land protected.
I don't really know, I would assume so.
The scenarios where they're not in ideal conditions, could they still just survive but not do as well?
I think so as well, because like the trees themselves are pretty hardy.
Yeah.
The main issue is like
The
growing
seedlings.
these trees, yeah.
Yeah, it's like Joshua Tree National Park won't be an ideal place for seedlings
Yeah,
if these projections occur.
because the trees themselves are really hardy, but then like another issue would also be like forest fires, all that. It's like the adult mature trees that are actually strong, how probable could they actually be there, have all this like infusing hazard on them.
How do you think the recent grades past 2 years have impacted their germination rates, taking, so on?
I would assume it would make it easier for them, but it'll also be kind of dependent on if they were blooming around that time, because they had like a very specific, like, I think it was 2 to 3 months, like, period for trees to actually like take. And then it was very specific, like the seedling had to be about like 20, I think it was 20 centimeters, to see if that would actually fully germinate or not. Because around that time, a lot of them died off. There's this really weird study I was reading, it said like 90% of the seeds were fully germinated and they were taking, but then in the next year, only like 5% of them fully survived to grow.
After a huge rain event?
I think it was after the brand, yeah.
Oh, wow.
With the climate change, is there a possibility that nature will start migrating to more favorable conditions rather than dying out?
I don't know. Cause I think the main issue is just like actually having them like migrate to those places, like relocate them there.
Great, we'll give, oh, one more question?
Do you think in the future, like you said, there are talks about indigenous communities having more of a role in relocating or germinating species in their preferred climates? Do you think that's gonna be a larger talked about subject in the next 20 years?
I think so, yeah, I think, cause I was watching this video that the Western Reservoir Act had on their thing, and the whole thing was kind of pushing on including more ethnobotany and actual plant communities, that way it's giving a voice to people who are like actually indigenous to the lands here. They can have like a voice to actually speak out on their opinion on the plants, and I think it's very empowering towards them and also for the body community. So I would.
Cool, give it up for honor. Cool, our next speaker is David Banuelas. David is a first generation college graduate with Indigenous and Hispanic roots. His early experiences in national parks led him to pursue a PhD in ecology and evolutionary biology at UC Irvine. During his time at UC Irvine, David worked closely with the Newport Bay Conservancy and the Big Canyon Project. His research is particularly focused on enhancing restoration efforts by paying close attention to mycorrhizal fungi.
Tip forward is, down is advanced.
Good evening, everyone. Really excited to be here. Slides there.
One second. It's full screen, here we go.
Alright, perfect. Well, first I wanted to point you to Big Canyon, which I think most of the people have been there before. So I'll talk to you today about some of the restoration that goes on there. And I also just want to give acknowledgements to Gabrielino and Quiche Native Americans, who are the traditional stewards of the land. So before I talk about restoration, I do want to introduce two kind of mycorrhizal fungi, which are arbuscular mycorrhizal fungi, which is going to be in blue in this talk, and then ectomycorrhizal fungi. And so ectomycorrhizal fungi really have the fruity bodies that you see down here below, so these two symbols, are muscular mycorrhizal fungi, have these like circular structures called arbostoles. And then down in the red here is gonna be ectomycorrhizal fungi, which have, fruiting bodies, which you see. And, my first time doing a fungal foray, when you look for fruiting bodies, was with, Joanne and Ron, a few years ago, so, that was a really cool experience, and so that kind of led me to do my research on, fungi. Alright, so first thing I wanna talk about, the trees today that we're gonna, part of the research is called dual mutualists. So they actually have two types of fungi, arbuscular and ectomycorrhizal fungi. And the advantages is they have more nutrient uptake, pathogen protection, there's a whole wealth of different advantages of having two types of fungi. And so that's what it looks like on a microscope. And so you have ectomycorrhizal fungi kind of like forms this thing called the arctic net. And then you have arbuscular mycorrhizal fungi, which is really in the root cells. And so basically what we did down here in UC Irvine was you take a little teaspoon of soil, you put it through a PCR machine, and then you put it through a thing called Illumina sequencing, and then you get actual richness and diversity and also the abundance of fungi, just from like a small teaspoon of soil, which is really awesome. So, is most people familiar with arroyo willow, right? We've seen them up down here in San Diego Creek. And so down in Big Canyon, the big problem was there was not enough arroyo willows and there was more resilient pepper trees, which is really overcrowding the species, and the arroyo willow was really impacted. So the Big Canyon project is basically like this huge $2.2 million project that occurred in 2020, and it basically was to remove 8.2 acres, 8 acres of Brazilian pepper tree, which is the invasive species. And really, if you look here on the map, the blue areas are just the arroyo willows on site. So the red area represents where they completely cleared out the Brazilian pepper trees. And the blue areas where they kept arroyo willows called selective removal. So full removal is where they remove every single Brazilian pepper tree in sight. And then blue is going to be selective removal, where they just basically kept the arroyo willows on site and try to minimize damage. And so what that looked like in the beginning of the project, you see this huge monoculture of a Brazilian pepper tree. And then if you we move into October, and this is about like 40% completion. They're basically just leaving their arroyo willows. Then if you look here, 2021, that's basically when the project was completed. So you can see they completely removed all Brazilian pepper trees. So the hypothesis we had around Brazilian pepper trees and arroyo willows was that these fungal communities kind of coexist together, but when you introduce the disturbance, there's going to be some detrimental effects. So our hypothesis here was that both types of fungi would be really negatively affected by the disturbance, because you're basically coming in with tractors, chainsaw crews, a whole bunch of disturbance, and that usually leads to a decline in both types of mycorrhizal fungi. So again, the red is going to be ectomycorrhizal fungi, and blue is arbuscular mycorrhizal fungi. But for today, we're just going to talk about ectomycorrhizal fungi, which I think are a lot cooler because they have fruiting bodies. So when we looked at selective removal, and again, that's like those blue areas on the map where they basically just left their arroyo willows on site. You can see here the green box represents following the removal. So you can see there was a significant decline. So if you look here on the box here, the y-axis is going to be the richness of ectomycorrhizal fungi, and then the x-axis here is going to be before and after. So you can see here the probably the median is around like 8. So for every scoop of soil, there's around 8 species of ectomycorrhizal fungi, and that declines to less than 5. So it sounds like not a big thing, but it's a big deal because these fungi are really important for plant function. And so moving forward with the full removal, where they just removed all the pepper trees, we have the same hypothesis that there would be decline in both types of fungi. So pretty similar to selective removal, but we'll see what happens. So surprisingly, ectomycorrhizal fungi actually increased, which is a big significant difference. And you can see here, before removal, the median was around like 3, and then after removal, the PD was basically above, between 5 and 10, which is a huge increase in fungi. And so that really puzzled me as a grad student at UC for a long time. But looking at the different species of ectomycorrhizal fungi that contribute to that, if you look at these two species here, Geopora, which is the genera, these two species really increased. So when you remove the Brazilian pepper trees, these two species just like they love it, they liked it. And so part of the reason why the full removal here actually did better than selective removal was because in full removal, they actually stockpiled the soil and then they treated it with different soil amendments, like organic soil amendment. They also added fungi to the mix too, but that didn't come up in our sequences. So or basically what we think happened was because we added a lot of these soil amendments, that actually increased the mycorrhizal fungi, particularly these Geopora species. So when you think about implications for management, you really need to go into the selective removal and add some of the soil amendments because they didn't do that. So instead of just leaving the royal willows to fend for themselves, you got to go in there and actually add some ectomycorrhizal fungi or the soil amendments. And you know, the only way you can do that is through soil inoculate, which is basically you can go and collect soil from like one of the local willow areas, and then you can actually bring it into the project area and kind of reintroduce that fungi into your restoration project. So 3 of the top species that we really have that Aurora willow really likes is called Tomentilla, and it doesn't actually have a whole fruiting body, I wish it did, but it looks really cool on the microscope. And then this is a whole Banuelas, which has a really nice fruiting body, looks like an orange peel. But my favorite one by far is Geopora, and it looks like a globe, so it's kind of like a geo. And that species is really important. And when we did the research, what you find is all throughout the world, anywhere there's a willow, a Salix genus, they're gonna have Geopora with it. So there's a thing called a polar willow, and there's also called a Mongolian willow, and those species have Geopora. So anywhere you go in the world, Geopora is gonna have the same kind of partner with them. But why is that important? So when we started looking at iNaturalist data, one of the oldest observations for Geopora was in the San Nicolas Island, which was done by whoever Newtonian is. But they actually collected the species and then they barcoded it, so they sent it to a sequencing machine similar to writing with soil, and that added a database. And then when I did my study, I was able to cross-reference it with what they found in San Nicolas Island. So anywhere there's a willow in the world, there's gonna be Geopora. And that's important because Geopora is really helpful with salinity and heavy metals. So willows are really resilient because of their fungal partners. And one thing I was really trying to do as a grad student was try to go find a fruiting body, which was super difficult. Even in the 2 years of rainfall, I couldn't find it in Big Canyon at all. But Randall English, who's our conservancy partner we have, he found a fruiting body last year. So I definitely implore you, when you're out there in the field, definitely take any pictures of mushrooms you have and upload it to iNaturalist because I might just be looking for it. So you can see here, we actually have a recording right here at Newport Beach, across the street, across the bay from Big Canyon. But that's Geopora stiposa, and I implore anyone to challenge it and try to get a better ID. But really, it's important that we collect these fruiting bodies and send them to get barcoded. So we have a DNA reference, and then when anyone in the world, whether you're in Mongolia or in Australia, and they collect soil and do the DNA, it will match into Geopora. And these species are super important because they're gonna help with climate change, like salinity, heavy metals, which are really detrimental to the trees. So Geopora is really important. So I'd like to thank my co-authors on this paper. You can also read the article, and it's open access on restoration ecology. And I'd like to thank particularly the students who worked on the project, also the Quiche Native Americans who also came and did the cultural monitoring for the Big Canyon project. And also my advisor, Kathleen Treespeeder. And also I like to thank the West County Native Plant Society because they paid for me to go to the conference a few years ago. And that was an amazing experience. Like I got on a plane with everyone from the chapter and everyone from Big Canyon, and so it was a really awesome experience. And I also met my wife here today, who goes kayaking with me and helps me look for some of these mushrooms. So, yeah, with that, I'd like to say, any questions?
Do you think the fact that, pepper trees being removed, contributed to the health of minority? I mean, obviously, it's not, bad, but couldn't that also have contributed to, in addition to the soil amendments? The fact that big bad guy is gone, make them happy and flourishing.
Yes, yeah, definitely, and so one way that we can kind of like have evidence for that is because there was more diversity of geopora with the salients beforehand. And so also another thing I didn't get into the talk today is the arbuscular mycorrhizal fungi, so the Brazilian pepper tree might use arbuscular mycorrhizal fungi more. And because they're so intermixed with willows, the geopora might just be there. So the way to test what you're asking is you have to go again to the project site and then also compare with the reference willow where there's no invasion. And then you can really see if geopora is positively correlated with the willow. And then you also need to go to the pepper tree site again and see if geopora is there. So the short answer is yes, but you probably need to go back again to see if it's persisting, if the effect is persisting, or geopora was just occurred in higher abundances as a result of the way they treated the soil. Yes.
What amendments do you recommend using? If I'm digging my own garden, and remove a plant tree, what do you recommend adding? Kind of soil methods.
Do you eat crabs?
Do I eat crabs?
Like seafood?
I do like seafood, but not.
Well, one of the ones they added was chitin. And that's like from a crab amendment.
Okay.
I always thought that was pretty fun. But apparently, chitin really feeds the mycorrhiza. And they have like enzymes, specialized enzymes to break down that chitin and then provide nutrients to the tree. So I wouldn't say crush up crabs from your household, but like they sell a part of it.
Really?
It's just called like chitin or something. Oh, chitin. If I follow this QR code, and look at the paper, then you can, I have all the amendments listed.
Sweet, thank you.
But I recommend chitin. Yes.
You can buy a product called crab shell meal at some of the garden centers, I know one. That would probably be extra for that. Okay.
Say that louder, Ron, please.
Oh, you can buy an amendment, a garden amendment called crab shell meal in larger garden centers. And it would be probably enhanced microarray development. I know when you remove the soil removal and you had to bring back soil, did you inoculate that soil with that same genus of, or did it just come back naturally on its own? I know you inoculated, the soil went back in.
So,
And my second question is, where did you get that inoculant from?
right, it's called a product called fungi perfecti, and I don't remember.
Okay,
So, one of the big things in the field is that the commercial inoculums tend to not work.
right.
Because you really can't just make a specific fungi inoculum for every situation. So you can't sell a national product of just specific ectomycorrhizal fungi. So when you look at fungi perfecti, it has 8 different ectomycorrhizals, like Boletus, Russula, really popular stuff. That's like kind of for pine trees, things like that. So they don't have anything specific for willows that includes a geopora, but based on geopora's relationship with willows across the world, there is no commercial inoculum with geopora. So that's why, if you also look at the paper too, and a big theme in our lab at UC Irvine and the Tree Seeder Lab is that commercial inoculums are usually not worth the effort. Although they use widely, it's more collecting soil and using a reference inoculum usually most of the time will do a lot better. So it's just about really understanding your local conditions and then, so that the commercial products tend to not be used so specific. Yes.
Is Geophora specific to willows, or is it also found in association with other like riparian plants?
So chapter 2 of my dissertation, geophores and the redwoods too.
Oh.
So I looked at redwoods doing the same sequencing, and you find geophore in there, so it's pretty common. Genera, like it's with a lot of trees, but specifically willows is where you find like beneficial effects. Yes,
Do we know if there are willow groves locally with your kind of salix that have other plants with them? And geophora and some other fungi are even much more abundant, not so abundant with the pepper tree, but if it's with something else, maybe it's even more robust, and therefore grabbing that soil for remediation could even be more productive.
yes, so definitely with other riparian trees like alnus. And other, what's another one? Alnus, any riparian tree that has a community with salix will be a more diverse mixture of different trees. So if you can go to San Diego Creek, there's like 3 types of willow. I think there's a black willow, a royal willow, and then there's also alnus, which I'm blanking on the common name. But yes, if you do go to a more diverse native community, you will likely have a different, higher prevalence of the beneficial fungi. But you really don't know which is gonna be beneficial, you just gotta throw it out there, and then when you get to this results, then you can like, yes.
So you're measuring species richness of like different subspecies of it? And where do you believe that that comes from? Like, is it different breeding that gives you more diversity? Or is it like introduced from ambience? Or do we have any theories on that?
Well, some fungi are like when they have a free body, the spores fly in the air, so they can disperse across large distances, and that increases the genetic pool. So the way I did the sequencing is just through DNA, so you just know like what's the genera. But if you wanted to do like a 23andMe, you could do it, and then you could say like this geophorus cousin lives down in Big Canyon, and then, but they're not related to this other geophora, they're a different clan.
So
So,
you got to genus.
well, we get to genus and we get to species for some of them.
Okay.
But you could have 8 different geophora genera, but they're 5% different. It's a 95% similarity.
Right.
But if you were to do RNA sequencing, which is a different type, then it'll tell you exactly like 23andMe, and you'd be like, well, you know, you could be like, Luke is my father, or you know. But yeah, you can do that, and with a little more money and like precision, yeah, you can tell who's what fungi are related to who. And then you can also tell what genes are in that fungi, and then it'll tell you what they're really doing, like salinity genes, heavy metal genes, and that stuff. We'll give it up for David one more.
We are gonna have our last speaker for the night. We are gonna have Emily come up. Emily Fitzpatrick goes by she, he, they pronouns. They are a 5th year, Cal State Long Beach student pursuing their BS in biology and a minor in environmental science and policy. He joined Amanda Fisher's lab, he joined Dr. Amanda Fisher's plant evolution and systematics lab in 2022 and began working with Dr. Fisher and his honors thesis project on Hesperocyparis orbiceae in 2023. She currently plans to work in conservation and restoration with a focus on scientific writing and communication. Well, and your microphone's on the stand.
Okay.
There it is. Okay.
Just hit slideshow.
It won't show up on Zoom. Yeah, it's there. 1 second. Soon. There we go. Meet back on the menu. Wait, where's Zoom? Oh my god.
Sorry, stand by.
It's late.
It is late. I just got back from vacation. It's always, it's the other one as well.
Yeah,
Thank you.
yeah.
Thank you for your service. No notes. And we should be going.
Okay, hello, I'm Emily, so I'll be talking to you about Dakota Cypress, SRO Cypress Forest and how it's being managed in the Queensland National Forest, specifically in relation to its relationship with fire. So, starting off, this has a fire adapted morphology. The species has surrounding cones, so they release their seeds when they are exposed to fire. And this post-fire environment also provides a really great germination site for the seedlings because there's reduced competition from other species. So they have the best chance of surviving and continuing to grow in the area. As far as its conservation status, its California rare plant rate is 1B.1, so that means that the populations in California and elsewhere are considered threatened or endangered, and the over 80% of these occurrences are also considered to be at high risk. The IUCN recently upgraded it from vulnerable to endangered in 2013, and despite this, it is currently not listed as threatened or endangered on a federal or state level, even though it is something that should be considered for listing on the state level. As far as its distribution, it's found in Southern California and Northern Baja. There is a large population at the Irvine Ranch Conservancy that I did not consider in this project because I'm working with the Forest Service to provide the data for management. So all of the populations I'm talking about are in the Chimico district of the Cleveland National Forest, so we can get them that information for management. As far as the life cycle of the species, it isn't able to reproduce until it reaches about 7 years of age. And at that point, even though it is reproductively capable, it is still fairly sporadic until it reaches about 35 to 40 years where it has stable regular reproduction. And cone production can actually continue to increase until it reaches about 90 years of age. So as far as seed cone development, this whole process from where it's open for germination on the very left to where you have these woody cones on the right can take up to 2 years. So reproduction for the species is a very long-term investment. So in terms of conservation, we have some concerns. There was a 2010 management plan and they determined the main concern to be fires, specifically fire frequency. If you have a population and it gets a fire going through it, everything gets wiped out, you'll have some seedlings that come back. If you have another fire in 4 years, nothing's coming back because there were no trees capable of reproduction. So fire frequency is the primary concern, and with climate change, wildfire risk is increasing due to drought conditions, and this species isn't really very adaptable. It isn't mobile with its dispersal method. It isn't really able to change where each location is, and we actually don't even know its genetic diversity very well. We presume it has a relatively low genetic diversity because when compared to its closest relative, which is an island endemic found on a single island off of the coast of northern Mexico, it had lower diversity in the populations that were compared to that island endemic. But that was only for populations in Baja California. We don't know for the full range of the species. So we are assuming it probably has low adaptability to climate conditions. Now, looking back at fire frequency and fire return interval, that being the primary concern, we want to have a solid understanding of that, and that's really developed over time in the literature. So back in 1987, this study used field data, and they found there was full reestablishment of a population at 36 years following the fire. If there were drought conditions, however, it took longer, it took 40 years. So they said, okay, 35 to 40 years should be all right for these populations. But in 2006, there was a new study that came out that found when they looked at these populations, anything less than 40 years caused a decline. And so they said, okay, we need to have it at least be 40 years. But they then referenced that 90 years for continued increased cone production and said it's probably ideal for it to be longer than 40 years so that cone production can continue to increase. However, in 2013, there was a new factor considered in a new study, and that was whether the fire regime was homogeneous or heterogeneous. So a heterogeneous fire regime is very patchy, so that basically means some trees can get burned, some trees are okay. A homogeneous fire regime basically burns the whole area. And they found that if there was a heterogeneous fire regime, 17 years is the minimum FRI. And that's because that allows for more demographic variability. There are still some mature trees that are reproductively capable, and the next generation is also coming out. But while that's really great information to have, that is very difficult to manage for, because whether or not a fire is homogeneous or heterogeneous, or the interval that fire frequency is happening in, depends on landscape factors, geologic conditions, climate conditions, a bunch of things, fire management practices, and that's something that you can't really create a very broad plan for, because it can be pretty specific to each location. So with this project, we have two research questions. The first being, how has fire impacted these populations in terms of size, demographic structure, and location? I hypothesized that there would be few individuals at these populations, and we would also have low demographic variation, and therefore we got high risk of extirpation. We also looked into whether the species is fully reliant on fire to germinate, which I won't really be talking about, but if you want to ask about, you're welcome to ask about that later. And for the data we're looking at, we haven't yet surveyed CNF1, but we've surveyed 2, 3, and 4, and we're going to be looking at that data. These sites are all about 1 to 1.5 miles from each other, and CNF2 and 3 are above the ridgeline, while CNF4 is at a lower elevation and a bit more sheltered behind the wind. As far as methods, we gathered site characteristics at each location. We took the height, diameter at 10 centimeters, and the number of cones for each tree. And then we used the height and diameter to classify each tree as either a seedling, adult, or mature adult. And that was based off the 2010 management plans classification system. Although we combined some categories to generate those 3 categories rather than 7, because we wanted to focus on the reproductive capacity of the individuals. So before we start looking at each individual population, if we look at this fire history map, you can see that there isn't a huge amount of overlap in terms of which fires each population has been exposed to. There's some, it's not a large amount, and they're actually also typically found on the edges of where the fire regime is. And the edges are actually where it's more likely to be a more heterogeneous and patchy fire distribution, so that's good. So for CNF2, you can see that there were no seedlings. There were 3 adults with a total of about 1,700 cones, and then 2 mature adults with almost 2,000 cones. There were a total of 5 individuals which matched the management plan, and the last recorded disturbance was in 1979 with the Paseo fire. We think that that's probably when all of these 5 individuals, their seed cones were open. We aren't sure why it's split into 2 groups here. We're thinking it might have to do with maybe germination was like staggered depending on environmental conditions, not sure. Or there may have been other factors related to the mowing, which I will talk about closer to the end. And yeah, that's it for that slide. For CNF3, they actually had a large number of individuals because of the 2017 canyon fire. That caused there to be a bunch of seedlings, and then there are also some young adults, but these are all producing at a very low capacity, so this particular population isn't really set up for success if there is a fire that occurs relatively soon. At CNF4, we saw 4 adults, 4 mature adults, and what's interesting here is that in 2010, they had 18 adults, and we don't know where the other 10 are. We don't know where they went. So that's something that we're curious about looking more into. And something else to note here is, especially compared to CNF2, there are very few seed cones being produced here. On CNF2, you had almost 3,000 with just 2 mature trees. Here we have 4 mature trees, and yet we have 37 seed cones. And so I'm thinking it might have to do with the location difference. Because things are wind pollinated, CNF4 is more isolated, it's more sheltered from the wind, so I'm thinking maybe the pollination isn't really able to get going as much. But that's also something that we just talked about yesterday, so I'll maybe have more detail on that in the future. As far as population viability overall, both CNF2 and 4 had very few individuals, but they are mostly adult mature trees. And CNF was a pleasant surprise at having a high number of individuals. And all sites actually had pretty good variation in terms of their age class, all had at least one mature tree, all are reproducing at some degree, so that was pleasant to find. Okay. And we think that CNF2 and 4 are most resistant to extirpation, especially CNF2, because they have multiple adults and mature trees that might be able to persist after a fire if they aren't fully burnt down. CNF3, however, like I mentioned previously, is at a sizable risk of extirpation if there are those fires in close succession, because it currently has low reproductive capacity. And the historic FRI is 15 years on average after looking at the fire history of that location. So that's something that should be considered when managing the fire in the area. And talking about fire management, thinking back to that fire history slide I showed you, where we all didn't have a huge amount of overlap between the fires they are exposed to. Again, these are 1 mile to 1.5 miles away from each other. So managing on a highly specific scale like that is very difficult for the Forest Service to do. And something that we found on our most recent fieldwork session, actually in late May, was that they had gone in and mowed CNF2 and 3. And what I mean by mowing is that these wood piles here are branches and occasionally trunks from mature and adult trees. The seed cones are also there, which is good, so the fire goes through, there's still the next generation. But cutting down the mature trees, like from this high individual that was producing the most out of all the trees we surveyed to a stump, still reduces the reproductive capacity of that population overall. And that's also a concern when you think about the genetic resources of the species, because that's something where we don't really know what it has in terms of genetic variability, and you could be losing a lot if you lose even a few trees. So, while it's really important to mitigate fire, because fire frequency is a concern, doing so should be done with consideration for, are they producing a large number of seed cones, what kind of genes do they have, because that's really a big issue of conservation of the species overall. As far as moving forward, we're gonna go back and get some more data and look at CNF1. And if anyone wants a project on genetic diversity, please consider doing the species and doing the full range rather than just Baja, that would be great. Otherwise, I'd like to thank the Cleveland National Forest Service, especially Lauren Pond, the Embedded and Peggy Kaiser Scholarship Award for helping farmers, OCCNPS for letting me hammer to you all tonight, my fieldwork assistants, Rachel Jake for their work below, and Dr. Sugar for keeping this on this project.
Fire, telling the Forest Service to cut down the cypress.
I don't know, I called them and I left them a voicemail and I think I missed their call back and I also. So I tried to get info, and we just, 2 shooting stars in the night. So I don't know, I'm thinking about calling them again, but.
Do it. It's lack of education too, I think.
Well, they did actually, they flagged at CNF3, they had flagged a lot of the individuals actually to avoid cutting them down, but it was mostly juveniles, and that's great, that they're marking them and avoiding data. But then they also cut down like 3 mature adults, and I was like, well, those are good to have. So, yeah. Yes.
But it would seem to me, even if they don't cut down a lot of the mature trees, if they were changing that understory, there's gonna be a conversion of that to other woody species that might outcompete the Ducati. Would you agree with that? The type of conversion of habitats is a concern in the building.
That is a good question.
It could just return to chaparral.
Yeah, I have bonus slides that kind of have pictures for that. So, like for CNF2 beforehand, you can see that it had like a lot of invasive grasses. Afterwards, it was mostly native species, except for, I think there was like invasive wild rye, but I'm not 100% on the field ID. But it did seem generally like it was more native species than it was previously. And for CNF3, it was similar. It was a lot less invasive grasses, so I think it was actually probably beneficial. I just have concerns over specifically their like pruning and such of these trees, cause I didn't really see the benefit of it in terms of reducing fire risk. Instead of just leaving them there or doing some sort of seed banking effort or something like that. Yes.
What's the lifespan of the seeds, especially for the mature plants that got mowed? Like, how long will those seeds last, just kind of in the cones?
That's a really good question that I don't know the answer to, but I want to look into that. Yeah, you mean specifically on the tree or just?
Just the ones that they mowed, because my concern is like if fire doesn't go by, they're just gonna die.
That's actually, okay, that reminded me of something, so something that we. Go to the second question, because that is related. So we initially were going to do, like, take cones for every population, like only 5, and count the number of seeds in each cone. And so we got some cones back in November, I went to count them in February, and then half of them had opened randomly just sitting in a lab at 80 degrees for a few months. And I was very perplexed by that, because everything I read had said that that would happen, you know, so we actually can't use that data really for anything because it got all jumbled together. But that shows that if they're removed from the tree, maybe they'll like just release on their own. The only issue with that then, since it's not linked to fire, is then that's not really a good germination situation for them to be in. So they're probably still not really gonna do well or do anything significant. And there's a really high rate of herbivory in terms of the animals that eat the seeds. So that's, yeah, the outlook is not great.
Any chance the foresters can do a prescriber in that area?
Yeah. You can talk to them.
Call up Lauren.
Give them the- Any other questions? Okay, thank you.
Oh, there's one question in the chat from Mark Sugars. Which of the sites you looked at was the one that the Urban Ranch Conservancy worked on several years ago? I don't think that was included.
No, we didn't include any urban ranch areas. I will say CNF3, I forgot to mention, it was planted by the Forest Service either between 1960 and 1981. We don't have a solid date on it because we just don't have a solid date on it. But it is doing relatively well, I would say. I think that's a good move. So they're like human intervention is definitely a good thing. Urban Range Conservancy is doing really great work with the species, I would say. They have a lot of individuals up there. But I don't have data on that, so. Cool.
Give it up for Emily one more time. Cool, and that was the student night, so thank you for coming out. I will say there are some Salvia spathaceae in the back, hummingbird stages I brought from Irvine Ranch Conservancy, so please take them. I don't want to take them back. So if you'd like to take an additional one, please take an additional one as well. So thank you for coming out. Oh, also, there are some books in the back by the cookies and everything. I don't know if they're all gone, so never mind. Thank you. Yeah. Thank you. Yeah, I'll email you and speak about getting on this.